Hello everyone, this is Anuja Misser. Today I'm going to present my topic for grant proposal, which is a study investigating the impact of diabetes on male fertility. Diabetes mellitus has been affecting our body in various ways. This study will focus on one of the more important but less discussed aspects of this issue. In recent years, studies have found that the high glucose level produced in body accumulated in blood during diabetes, which also further interacts with the germ cell environment, is one of the major causes of infertility in humans. It has been observed that excess sugar level in blood causes distortion in sperm structure and even interrupts in its normal functioning by reducing the mortality rate. This study will focus on males of age group 25 to 50 years in the United States. For this research, the dependent variable will be diabetes and the independent variable will be male fertility. Various clinical experiments will be performed and correlation between diabetes and male fertility will be observed. The study regarding diabetes related male infertility have been and will remain an important topic to uncover the actual mechanism. The study will certainly help forward for possible interventions regarding metabolic clinical analysis. So this is the outline for my presentation for today. Okay, so starting up with the introduction. What is diabetes? Diabetes is a condition in which more than the required amount of sugar stays in the blood because of the inability of pancreas to secrete enough insulin to regulate the blood sugar level. Glucose is an important source of energy, but its excess can impact our body function very badly. In recent years, a new cause has been identified which is linked to infertility in males and is still under research. Elvis et al. In 2013 informed that diabetes mellitus is considered as one of the greatest public health threats in modern societies, not only in the Western developing countries but also in many developed countries. The increasing incidence of diabetes worldwide is supposed to inevitably result in a higher prevalence of this pathology in men of reproductive age, and subfertility or infertility associated with diabetes is expected to dramatically rise in coming years. Male fertility is caused by inability of sperm to function in a desired way, which may be due to sperm motility, decrease in the sperm count, abnormality in the chemical constituents inside the sperms, etc. Diabetes is believed to indirectly affect the motility as well as the DNA present in the sperm cell. Pasmezzo et al. In 2016 suggests that subfertility constitutes an important health issue that concerns both the developed and the developing world. According to this study, approximately 16.7% of couples come up against with the problem and throw on doctors in order to receive the appropriate therapy that will lead them to in pregnancy. It is important to note that an obvious reduction in the quality of semen has been observed during last 50 years. This study helps to understand the relation that the past years have significant increase in diabetes and infertility cases are rising up with the same pace in around the same years as diabetes. According to Abassi Homrosi et al. In 2019, oxidative stress experienced by the diabetic patients caused due to the unbalance in the production of reactive oxygen species and antioxidant defenses inside their body is one of the major causes contributing towards infertility. It may also slow down or pause growth of sperms to its final and functional stage. By experimenting and clearly observing the above described factors that I said to take sperm fertility, the impact of oxidative stress over the other physical factors on the sperm counts can be analyzed. The other observation that can be considered to observe sperm activity and abnormality include the ability of sperm to undergo capacitation, A chromosome status, membrane integrity, interaction with oocytes, mitochondrial activity, DNA integrity, etc. Keeping these factors into consideration, Visconti et al. In 2021 found that it is important to notice the molecular level to observe what factors are causing sperm to become non-functional at a given time. According to a study conducted by Elvis et al. In 2013, it is confirmed that problem in spermatogenesis process caused due to diabetes has a great impact on endocrine control as it impairs penile erection and ejaculation in males. Therefore, male infertility may become more widespread as diabetes rate increase. This study will investigate the impact of diabetes on male fertility and the extent of level of damage it causes to the structure of sperms, making them to be non-functional. Previous research and studies based on this issue will be reviewed in the study and a correlation will be obtained on the actual impact. Diabetes may also impact the epigenetic change during spermatogenesis. So, as described by the study of Jiao et al. In 2018, the sperm can also contribute to the inheritance of diabetes to more than one generation, causing chances of infertility in their offspring. Jiao et al. In 2018 also suggests that the process called testicular apoptotic cell death, in which the germ cells produced in males are destroyed by the programmed cell death happening inside our body, contribute to diabetes mellitus induced male infertility. It eludes an important molecular level process which involves microRNA 34A as a pro-apoptotic RNA which targets serotonin 1. Serotonin 1 is a protein that provides protection against various complications of diabetes mellitus. This target action causes apoptosis of testicular cells. In the experiments conducted by Pelvinkova et al. In 2017, the testicular tissue of diabetic males showed significant tissue damage, disruption of meiotic progression, and changes in the expression of genes encoding proteins important for the spermatogenesis. Pet Paternal diabetes altered stem quality and expression pattern in the test in the offspring of two subsequent generations. A study performed by Sengupta et al. In 2022 suggests that a drop in Leydig cells count was associated with the reduction theorem LH, which is luteinizing hormone. That this may partially explain the stimulating role of LH on Leydig cells in the laboratory setting. Additionally, diabetes reportedly affects the spermatogenetic cycle by impeding the FSH, which is follicle stimulating hormones. It impacts these actions on the Sertoli cells. So this study aims to contribute to the pre-existing research studies and to add up valuable points to correlate the two variables, which is diabetes as an independent variable and sperm functionality or male infertility as an dependent variable. It is hypothesized that if a male is suffering from diabetes mellitus, then he would have less sperm counts leading to infertility. This issue that diabetes might cause male infertility needs more attention as there is a lack of awareness in our society as to what extent diabetes can harm the male sperm and therefore make them incapable of reproducing. by themselves. There are surely many in vitro techniques developed by the science to like artificially give birth to young ones, but this technique is not affordable and available to everyone. Also, diabetic patients may also always experience infertility. Therefore, it is a challenge to identify the exact factors and the extent of damage caused by each factor, which may directly or indirectly link with diabetes. So there is a great need to be aware about the impact of diabetes, which sees no age, on the sperm functionality. This study seeks to understand the correlation between diabetes and sperm damage, along with the external factors involved in treatment that might help overcome this issue. Okay, coming to the materials and methods. So for this study, um, I'll be using a qualitative method approach, and will be conducting clinical experiments. So that will be the primary source of information that I'll be using for my study. And the independent variable will be diabetes mellitus, and the dependent variable will be sperm fertility. The sample population that I'll observe will be 200 males from the United States with the age group of 25 to 50 years, and obviously informed consent will be provided and no harm will be caused to the participants. And for the materials and procedures, I'll be performing 3 tests, which will be testosterone and cortisol analysis test, antioxidants capacity test, and the third one will be the semi-structure interview or the physical test that I'll be doing with the 200 male participants for the study. So this in the screen is the budget that I require for my study. So for the equipments and materials, I will be needing in total of $8,534 dollars and 51 cents, including all the freezer, centrifuge machine, water bath, microplate reader, ELISA kit, and some more important things that I need for my experiment. And to pay the gynecologist, endocrinologist, and the genetic counselor for my study, I'll be required to pay them in total of $285.45. And for the rest of the supplies that I need for my experiments, will reach in total of $31,410.27. So for my budget, I will require the total cost of $40,230.23. So for conclusion, several researches have been focused on the adverse effect of diabetes on male fertility and the molecular mechanism caused inside our body during diabetes that induce alteration in sperm structure and motility. But much remains to be discussed. Insulin dysfunction in male diabetes patients is scarcely debated in the interpretation of mechanisms which are responsible for male diabetes induced infertility. The study aims to provide an up-to-date, detailed, and informative analysis of changes occurring during diabetes that might cause sperm to be infertile. That will be analyzed using clinical equipments and will compare the samples with non-diabetic fertile male patients to clarify the subject with a greater understanding. This is the references I use for my study. Thank you. I hope you like my topic for the grant proposal, and I hope I was able to explain the importance of this issue. Thank you.